And okay, thank you all of you for joining to today's session of the Animal Genomics NF4 Special Interest Group, and especially our speaker, Christopher Tuggle, who will be introduced by Francesca Bernini, that is co-leading this group, as you may know. Before she introduces Chris, I just want to say that we are recording these meetings, and the meetings are always available in YouTube. So that's why I'm not accepting requests for recording the meeting. So please, Francesca, the stage is yours.
Thank you so much, Jose. And today is my pleasure to introduce Dr. Christopher Tuggle, who has a PhD in biochemistry and is now a distinguished professor in the Department of Animal Science at the Iowa State University. Dr. Tuggle is a leading researcher in functional genomics with a particular focus on swine genetics. His work has significantly advanced our understanding of the porcine genome, particularly through his contribution to the functional annotation of the animal genomes FUNC project. And he also played a crucial role in developing the SID pig model. I'm sorry if I pronounced it wrong. An immunodeficient pig line that served as a valuable tool for preclinical testing of stem cells, therapies, and cancer treatments. His research has also shown that pigs, due to their genetic and physiological similarity to human, can be a powerful model for transitional medicine. Additionally, also, his studies on transcriptomics have shed light on critical aspects of animal health, including immune responses to infections, reproductive biology, and feed efficiency. And please join me to welcoming Dr. Christopher Tuggle. The stage is yours.
Thanks, Francesca. Can you hear me okay?
Yeah.
Okay. Good, good. So I'm happy to present some work here today. If there are questions, just interrupt me and we can discuss those. It's a small group, so I'm happy to be informal. What I decided to talk about today is really shown on, let's see if I can get to the next slide here. Yeah. Just give you a little bit of explanation of what I wanted to talk about today in the 40 minutes or so. Talk a little bit about why I'm interested in single cell and why other people might be interested. I give an example of single cell RNA-seq and single cell attack-seq. So a couple of examples in pig immunity. And then a little bit about what we are doing in the broader agricultural community in terms of developing a single cell genomics group, and how we could actually get to a place where people could do it efficiently. And then I have a few slides on perhaps a discussion with your particular group, and where I see the needs of that especially perhaps could be interesting to bioinformaticians. So a little bit of why single cell RNA-seq. Bulk analysis is essentially one value for each gene for every replicate of tissue or analysis like that. The problem is that it ignores the heterogeneous nature of tissues, and single cell analysis can give you more information about the transcriptome or the epigenome of the individual cell types in a bulk sample. So, if you don't analyze at single cell level, you don't really know what's happening in the individual cell types. You don't see what's happening in a particular tissue with respect to space. And of course, there's a lot of spatial transcriptomics going on now as well. And when you have the ability to measure RNA or other things at the level of single cells, you can look for markers of cell type. You can estimate the frequency of cell type in the population if you have a matrix of gene expression by cell type. You can look at different cells in tissues across a process like infection or developmental time, and you can identify cells that are transitioning during that time. You can also look at differential splicing between populations. Differential splicing is clearly important in terms of matching gene expression and genetics. So that's quite important to understand what's happening in different populations. You can look at allele-specific expression patterns. So you can have different alleles expressed in a population of cells, and you don't know whether you're having monoallele expression within cell type, giving you a mixture, or whether you have different allele-specific expression in each cell type. So if you have the ability to look at each individual cell, you can distinguish those patterns. And then, of course, you can look at expression EQTLs at the level of single cells, where you have some cell types where you have an effect of genetics on the expression. You have other cell types where that may not be seen. And you may only see some of these EQTLs that are genetically controlled only with stimulation. So the ability to measure the gene expression within individual cells can add quite a bit of information. And the other thing that's of interest is for example, combining data that's already existing for tissues and trying to understand tissue-specific gene expression. So for example, we took the tissue enhancers that were reported from the Pan et al. Paper across the 12 tissues that they looked at. They identified enhancers based on ChIP-seq analysis, RNA-seq analysis, and so forth. And what we looked at is the immune-specific, cell type-specific enhancers in some recent data that we're preparing for publication. And you can see that depending on the tissue, some of these enhancers that were identified in the tissue are actually enriched in specific immune cell types. And so the idea is that we can take bulk data and refine it to look to see where the gene regulation is occurring, which cell types. And then finally, an example in the human field where how do we try to link directly to understanding quantitative traits and breeding. And there's an example of the paper about 10 months ago published on regulatory networks at the single cell level for brains, human brains. And they developed what they call the Brain Scope resource, which is basically looking at single cell gene expression, single cell regulatory elements, and putting together a model of what they call a linear network of cell type phenotypes. And so the idea is, can we understand what's happening relative to gene expression controlled by specific genetic variants that can explain across the entire tissue. What they're really hoping to look at is predicting phenotypes and prioritizing genes to understand quantitative traits in brain development and brain disease. And as you might expect, it's a very large number of people with an enormous amount of funding. So what we have to think about is how we might be able to do some of the similar kind of work without the significant funding that's here. And one slide I put in more recently is just what is the Human Cell Atlas community envisioning, and thinking ahead. One of the things that they're really pushing is this idea of foundational models of biology, where once you have a large amount of data on understanding the gene expression and the epigenomics of tissues in the level of space and multiple types of data collected, can you pull that all together into what they call this foundational model, and to try to understand the relationship between genetics and expression across these different modalities. And so this is quite a recent visioning paper from Human Cell Atlas. So what I'd like to do is just transition then to sort of the basic concepts of why, from the basic concepts of why we'd want to do that and what's happening in the Human Cell Atlas community, to actually how does it work. And so just a few slides on how you actually do single cell analysis. Hopefully you can see my pointer. Essentially, this is showing a microfluidic approach. There are other approaches, but the idea is that you have cells that you're trying to capture and read the transcriptome of. They are encapsulated with a bead, which is coming in from the bottom here. And then oil pressure is creating media droplets that contain one cell and one bead. And so within that droplet, you have a barcoding bead as well as the cell. Under particular temperature changes and UV light, you can break down this bead, and essentially what the bead has on it is oligonucleotides that will be hybridized with poly A plus messenger RNA and encode the cDNA with several tags, including a unique molecular identifier and a barcode for that particular cell. So let me just go on to the next slide and show you one other method, and then I'll talk about how that actually is, how those RNAs are mapped back to the particular cell. There's another method that's not microfluidic based, but basically sonication based, creating micelles and creating many, many thousands or hundreds of thousands, even up to 1 million of these encapsulated cells. And so there's some advantage to this new approach because it doesn't require any complex instrumentation like a microfluidics chamber, and it has relatively low cost startup. The other important thing is that they mentioned is the scalability, and we've used sort of in this range where you can look at thousands to tens of thousands of cells, but they can scale up or down from that sort of scale. And the advantage for what we've been doing is because we had quite a few samples, we wanted to keep our costs low. And there's some evidence that depending on what you're trying to do, as we'll talk about on the next few slides, 3,000 to 5,000 cells may be sufficient, and the costs are only hundreds of dollars, and then adding the sequencing costs to the individual samples. So there's two goals in single cell analysis from the point of view of scale and goals. You can start with deep profiling to try to find and document transcriptomes of all the cell types in your tissue. And that usually is going to require 20,000 or more cells, because one thing that we need to do is try to identify all cells, and you need to cluster even the very low abundant cells. So there may be cells that are perhaps less than 1% of all your cells. And if you want to really show the transcriptome of a small cluster like that, you probably need at least 50 cells, if not maybe 100 cells. And so that's why we think 20,000 is needed. Now that can cost you anywhere from $1,200 to $4,000 a sample, depending on technology. And then you have to add sequencing on top of that. So that's a low throughput, but high information content approach. And then beyond just collecting the data, there's a huge effort, a substantial effort to annotate the cell types using field expertise. And that can be anywhere from weeks to months of analysis of these kinds of data sets. And in just an asterisk, it's difficult to verify that you actually found all of the cells. So that's, hopefully you're getting 99 plus, but you may still be missing cells. Then if you transition, once you have perhaps that foundational data, you might transition to look at phenotyping of cells across many samples. And there, as I mentioned, maybe 3,000 or more cells. And once you have a reference, you can very likely be able to map 95 to 99% of the cells in a sample with just as few as a few thousand cells. And as I mentioned, the costs are significantly less. And probably more importantly, it really decreases the field expertise if you have a reference that you can map your data to. But again, then the reference data set is needed. So just to give you an idea of the data processing, if we think about the overall, what you're trying to do here is, again, we're starting with a cell and a barcoded bead. The cells are then going to go through the process I just mentioned, where the RNA is going to be will align or hybridize to the oligos in the bead, and essentially the two pieces that you want to keep track of is the cell barcode and a unique molecular identifier, and that identifier is actually truly unique. It's a single base. It's a single time in every one of these barcodes. So that allows you to do some fairly unique things that you can't do with some of the bulk transcriptomics. So anyway, you show that you see a barcode for this kind of cell. The red barcode is only in the red cell, and green in green, and so forth. And then you have this unique molecular identifier, which is not going to be identical in the next cell. So just don't get confused about the colors. That this is not the same as this blue color. It's a unique. And so it's actually means unique. Oh, I want to go back one. So just to take you through this, you basically collect the first strand synthesis. So the RNAs all get those 2 barcodes. And then the libraries or then the cells are actually combined together, and the library is made as a bulk, because now the RNAs are all labeled. So you don't have to do anything further with individual cells. You just do a regular library as you would do for bulk. But then, of course, the crucial point is the demultiplexing. So with the bioinformatics, you can then identify all the transcripts that came from each individual cell. And then the UMIs allow you to collapse any PCR artifact transcripts that are just PCR duplicates of each other, because literally there's no other transcript that should have that UMI. So you can collapse it, and you get a little bit better analysis from that kind of an approach. So that's in a nutshell, the simplest way of describing how we can get the single cell RNA-seq data. So what I'd like to do is give you a couple examples of this, and what we've been doing and utilizing this technology. And Mehek Kapoor has been analyzing the data in my group. The data is coming from Yin Fang's work at the University of Illinois. And the purpose of this is to try to understand the gene expression during an acute PRRS infection. And if you're not familiar with PRRS, it's probably one of the most important animal diseases. Certainly, it's the most important viral disease in pigs. And I'll show you why we really need to better understand this, is that the challenge is really detecting the persistently infected pigs. So PRRS actually infects pigs, and you can get that antibodies detected fairly rapidly, but for whatever reason, and that's what we're studying, for a number of reasons, we think that the virus just persists. The pigs cannot get rid of it. And so the virus continues to be present in the pigs, perhaps up to several months, and can be infectious to other animals in the herd. So when you get an infected herd, it can be a problem for the producer for many months. And we have tools to identify the virus, and we can identify animals with the virus, but we don't have good ways to really early predict the PRRS persistence. We can't really tell with these tools which animals are going to be able to resist the virus and get rid of the virus early, and which ones are persistently infected with virus. And that's shown here. You have an acute phase that all the pigs will go through, but some of them will then end up rebounding or being persistent in terms of their viral titers in their blood. And we really don't understand the immunity of the difference between the persistent and the animals that can actually extinguish the virus. And we'd obviously like to study the immune system in these animals, but it's very complex, and we'd like to see if we can develop a blood predictor. So if we could predict the animals that are going to be persistent, we could try to provide more treatments to those animals. So that's our goal, is to try to find new tools to detect persistent infection. And so the idea here is to collect information from single-cell RNA-seq and bulk RNA-seq, then use computation to uncover if there are unique host cell types or changes in those cell types over time, and then use statistics to elucidate the cell composition and the gene expression response and the regulatory elements that's happening during infection, and try to identify differences between these two classes of animal, persistently infected versus animals that extinguish the infection. So we start with 4-week-old pigs, where we infect most of them with a specific virus, PRRS virus isolate, and then we follow every week a blood sample on every animal, and at the end collect the tissues. On the PBMCs and the lymphoid tissue, we use RT-PCR for virus to then characterize which animals are extinct and which animals are persistent. And at this point, we've been able to collect these two groups of individuals, not a large number in some of the groups. And the idea is to look at single-cell RNA sequencing of the peripheral blood mononuclear cells to try to move toward a predictive approach. And what I'll talk about today is the data from day 14, which is when all of the animals have virus and all of the animals are have virus in their blood and have virus replication. Well, I'm just going to jump directly to the results because this is just an example. We did collect about 50,000 cells across those animals where we had about 25,000 evidence for about 25,000 genes expressed across those cells. This is all cells and all genes, not just specifically monocytes. But what I want to talk about particularly today is just an example of one of the cell types where we see differences. So you may know exactly, I didn't talk about this, but essentially one way to visualize all the cells is a dimensionality reduction approach called UMAP. So each dot in this diagram is a cell, and you can see what the control cells look like. The cells from control animals that were uninfected, cells from animals that are persistently infected, which is down here in the green, and the animals that have cleared the virus are in orange. Combined, you can see, the colors indicate the different types of cells and how do we identify what kind of cells they are. Well, we have marker genes that we know should be expressed in certain cell types, and the colors of the clusters of cells are shown here. So in pink are up here at the top. So two clusters, cluster 10 and 14, have markers that are known to be highly expressed in monocytes. And so we identify the pink set of cells as monocytes. What's interesting is if you look at the control animals, they have much fewer monocytes than the persistent and the extinct animals. So that's one interesting tidbit right away. We see that monocytes have dramatically increased in the blood. So that's interesting, but what we want to look at is persistent versus extinct, not infected versus uninfected, because we can detect infection by using a PCR analysis of just looking for virus. But what we really are interested in is extinct versus persistent. And we had no differences in abundance between extinct and persistent. But what we do see is a very large amount of differential expression in, for example, monocytes. And this is showing a volcano plot of the genes that are differentially expressed. And the fold changes are not large, but we do see some very interesting patterns if we look at the Go annotation of these cells. Genes that are higher in extinct monocytes versus genes that are higher in the persistent, the monocytes from persistent pigs. If you look at the Go enrichment, you find key pathways that are really associated with animals that can get rid of the virus. And as you might expect, that these are very related to pathways that are known to be important for removal of virus or regulation of type 1 interferon. Type 1 interferon is one of the most important immune pathways that must be turned on if you are getting rid of a virus. Maybe what's even more interesting is the genes that are persistently higher or higher in the persistent animals are cytoplasmic translation and oxidative phosphorylation. And so these are known pathways as well for cells that are not responding as you'd expect, and especially things like oxidative phosphorylation, which is a pathway that tends to lead toward the incorrect kind of response to the pathogen, and a cell that is more anti-inflammatory and more regulatory. But at this point, at 2 weeks of infection, we really want the virus to be gotten rid of. We really want this kind of pathway. So this is as expected, and now we can look at the details and try to find what might be really specific there. And I just wanted to mention, I've only been talking about monocytes, but we see differentially expressed genes in every cell type, and we see differential abundance in a number of cell types as well, primarily in extinct versus control, but we do see differential expressed genes across extinct versus persistent for multiple cell types. And this is a summary of a very large amount of analysis to try to understand what's happening in each of these cell types between extinct distinct and persistent. So I really only showed you one piece of the window of what we have to investigate. But as you'd expect, the animals that are able to get rid of the virus have a lot of immune activation, whereas in the persistent, the cells are not responding correctly and are doing things like cytoplasmic translation and antigen presentation. So just quick takeaways. I think I'll go through these very quick. We see that there is some potential for identifying a roadmap for predicting outcomes because we see differential expression in specific cell types, and we see differential abundance due to infection. And so there's clear strategies based on analyzing this data. There's clear, distinct strategies of the immune response to animals that are persistently infected versus animals that could clear the virus. And certainly monocytes is one cell type that we're really strongly investigating. What we can also do is try to identify regulators of these pathways. And so one thing that we can do is look at single nucleus ATAC-seq and try to really study that. And so I have a little bit of data on that, which I'll go through very quickly. But Penny Yang in my group has been analyzing ATAC-seq, using ATAC-seq to analyze which parts of the genome are open and are predicted to be regulatory elements across these same immune blood cells. But we're doing this on healthy animals. So ATAC-seq identifies the places where there's no histone marks and the transposase can get in and digest the naked DNA. And so by finding fragments that are coming out of an ATAC-seq analysis, you can then predict the open regions of the chromatin. And so we started with PBMCs, we ran this analysis, and we did clustering just based on chromatin accessibility. And so this is the same kind of a UMAP, but instead of RNA patterns, this is open chromatin patterns. So where are the open chromatin in particular cells, and can you group them according to that pattern? And what's really interesting is that you can try to annotate those cells based on mapping the open chromatin to the nearest gene and assuming that that is an activity of that, or can measure the transcriptome activity, a transcriptional activity of that nearby gene. It allows us to link open chromatin data to gene expression data. So we're just looking at correlation. We have many, many cells, and that allows us to do a correlation analysis that's pretty powerful. And so the gene activity is simply, do we have a lot of open chromatin near a nearby gene? Then we're assuming that there's a high level of activity of that gene. And when you do that, you can then compare those genes that are active and try to identify what cell type they are. And what's interesting is if you look at gene activity measures, so the assumption is that the open chromatin is predicting something about the nearby gene's activity, you can see that it really does quite a good job. There's a few differences, but there's an amazing amount of correlation between open chromatin and gene expression. And just a couple of things, I just wanted to show that once you have all these open chromatin peaks, these regions that are open, you can then look to see which ones are cell type specific, and those are called differentially accessible. And you can find many, many differentially accessible peaks across cell types, as you might expect. And just to give you an example, this is a gene called PAX5, which is a B cell marker, and you can see the chromatin appears to be predicted to be open near the promoter of that gene, and it's closed everywhere else except a little bit in antibody secreting cells, which are a type of B cell. So I'm going to skip this. I'm just going to say that once you have open chromatin, you can then look at transcription factor binding motifs that can be enriched in that open chromatin and try to predict transcription factors and even regulatory networks that could be controlling the cell type specific expression within these cells. And because it's been published now at the end of last year, you can go there if you want to see some of that detail. And I'll skip that. So I just want to transition to some of the resource needs that we see and what we're working on. This is single cell research that can be done at a single lab level or a small groups consortium level, but we argue with democratization would really benefit agriculture as it has done biomedical science. So there's a number of things that are very difficult to do within a single lab. The biggest important difference or what's difficult to do is to have optimal cell annotation and gene regulatory networks, because it would really be useful to use everybody's data for that. And so what we've been working on is an approach that improves what the current state of work is in agricultural single cell analysis, where the problems are that the user provided metadata is the starting point, which is not findable, accessible, and other aspects of FAIR, but we'd like to improve that. And we really need community-wide resources to do that. And so just as a quick example, what's happening in metadata flow and data flow in plants and animals, there's two sort of different approaches that are taken by the plant groups compared to the animal groups. On the plant side, they're dependent on users submitting data to the databases, and then any kind of pipeline to annotate the data is a very narrow pipeline because it's dependent on paid curators at the ENA, at the EBI. It's an excellent approach, but the pipeline is quite narrow. On the other hand, the way that the FANG group has done the work, as many of you know, the FANG has asked some data submitters to provide rich metadata. And so the metadata is already available, hopefully available for most data sets. And the idea is that we really just need ways of annotation and automated annotation to get to databases that can actually process the data. So one thing that we've been doing and published this paper last year is that what we needed to do was create an automated pipeline. And so what we showed as a proof of concept is that we could develop an automated pipeline with developing new single cell metadata tools and be able to validate those through the FANG data portal into the Human Cell Atlas data portal and into Terra, which is quite a sophisticated and well-supported database in the human community out of the Broad Institute, where there's a number of tools that are available. And so Muskan Kapoor in my group then took the data all for some single cell PBMC data and took it all the way to Terra and showed that we could do, for example, a regulatory analysis using Gene3. So in the last few minutes, I wanted to just briefly talk about what we've been trying to do in terms of single cell research. And the first question is, what do we need to succeed? I think we clearly can take what's been successful from the Human Cell Atlas community, and we need global repositories that people can access and have common tools. We need to have community annotation of the cell types, and then we need to apply it. And that we're hoping that will really improve genome to phenome and genetic improvement. Of course, we need to identify additional things that we haven't thought of, and that's why we need to have some conversations. But we obviously need to figure out a way of obtaining the resources for these things. And so Agricultural Genome to Phenome Initiative is something within the USDA as a resource for grant funding. They also fund conferences, and so we applied for a conference grant, and that's what's happening here in a couple of weeks. The end of the month, we're developed a single cell oriented workshop within agriculture, and it's called Empowering Genome to Phenome in Agriculture, Building a Collaborative Environment for Single Cell Applications. It's being held immediately prior to the AGBT Ag meeting in Orlando, Florida. So we had a bunch of aims that we wanted to complete. One was a survey, and so we put out a survey to the community, and that went to 4,600 email addresses. So hopefully most of you are in AngenMap and hopefully received that, or maybe even in the plant phenotyping community or the ag biodata mailing list. And we had about 200 responses. What I thought was interesting maybe for you, for this group, is that one of the top interests from the survey was connecting biologists with data scientists interested in single cell analysis. And then the application of AI machine learning and single cell analysis. So I would say those two top interests really are indicating that, you know, the bioinformaticians really are popular with the data creators. So we also have had 4 online single cell field days, which are essentially an hour and a half where 2 or 3 people present their work, and then we discuss what could be done to broaden that work to additional people. And if I'll leave this up here for a second, it'll be on the recording. So if you would like to get the recordings of these 4 sessions, and you can see their titles here, this QR code will take you to the website within FANG that it's in the FANG bulletin board if you want to go there directly. And then if you're still interested in registering for the AGBT Ag meeting or the single cell workshop that precedes it, that QR code is there as well. But a week and a half from now or so, we're going to start on a Saturday evening, and Jennifer Clark is going to talk about how do we build a transdisciplinary community. And then we'll have a few talks by leaders in the field describing where we are in plant and single cell genomics, as well as where we need to be. We'll have breakout sessions on trying to identify and achieve best practices in describing, storing, and sharing data, creating common data environments so that we can do cell type annotation, and then how do we identify and benchmark pipelines. And then we'll have a group discussion on identifying how do we get this done, basically. And these are some of the questions that we think that we're going to address. What are the additional needs? How do we try to get government funding agencies involved, maybe through a white paper and identifying people to do these things? So I'm going to stop there for the first part. I just wanted to point out that Muskan Kapoor did the work on the pipelines at the end, and I mentioned Penny Yang and Mehek Kapoor. This is a long-term collaboration with Crystal Loving and Ying Fang at Illinois, and especially Jack Deckers at Iowa State. So I know I only have a few minutes, but I wanted to have some slides on maybe things that we could think about today. And I think we have a little bit of time after I get done, but I just would pose a few things that maybe we can discuss. I didn't cover spatial transcriptomics, and if you were interested in that, I don't do that. I really focused on blood. But can we think about data metadata standards? I think we have an excellent starting point with the FANG metadata standards, but how do we encourage use of these in the single cell projects? And then from the point of view of single cell data analysis, I know that NF Core has a single cell pipeline. It's only 10x, and it's only the data to get started on data analysis. So we really need to think about cell annotation pipelines, and how do we get to a point where there's a reference that people can use? Who creates this reference? Do we integrate? Who vets the data to create the reference? Who provides the reference? And thinking along those lines, what does the human community do? And setting up and performing a reference mapping is currently a very important issue in biology. So this is still a perspective just from last year. And I won't go through all the details, but just 2 things that I wanted to point out is if you have a reference, they're arguing if you have a reference, you can take a new data set, map it to the reference, and get an annotated cell type. And the advantage of that is that the reference is going to have unique and specific cell types with the same names, the same annotation, the same metadata. So that's very helpful because then you can then compare your data to anybody else's data that's been mapped to the reference. On the other hand, you may have a data set where there's a new cell type, right? And the way that that's usually identified is the uncertainty of mapping. So there's uncertainty scores, and you can say, all right, there's a cluster here that doesn't map to the reference data. So that's a way that we can discover new populations and then further iterate and improve the reference data. So this is what they're doing in the human community. It's just something to think about for our community. So there are other data analytical operations where we need a pipeline. Amazingly enough, there's not a lot of pipelines for treatment effects and random effects. For example, repeated sampling, putting animal in the model. They don't have that a lot in the human community because usually you're collecting a single sample from a patient. And they don't have multiple samples from a patient. So we found that there were very few pipelines where you could put animal in as a random effect. Integration of cell data is a big issue, cell data across samples. And for example, one of the problems with integration of data by merging data sets from different pipelines or different data sets is that you have to be very careful. If you mix samples to try to get rid of batch effects and try to correct for those batch effects, you may be getting rid of unique cell types or more subtle differences that you really are trying to follow and see in genetically different populations, in treatment effects, and so forth. So this is, again, a recent paper, and it's still it's a big issue in the human field as well. The other important thing that I am working on myself is how do we get to deconvolution? And I haven't described deconvolution, but the idea behind deconvolution is if you can come up with a data set or a reference matrix for predicting cell types within a mixture, you can then use what they call deconvolution to take all the bulk data we have and convert it to single cell data. So we need a gene expression matrix for most analyses. So one thing is, should we provide that, or should we just provide the data and allow creation of a gene signature matrix by the users? And I think, obviously, finally, we need to demonstrate the value of this in breeding goals. So I'll end there. Sorry, I went a couple minutes over, but I wanted to also mention that the ISAG meeting is happening this summer, and so the registration is open, and I would encourage you to attend, where the scientific theme is genome to phenome for new challenges. So I'd be happy to go back and stop sharing. But if people have interest, I can put up some of these slides if there's interest. I'm hoping that we can get a conversation started with. I assume we have a few minutes to further discuss things. But I'll end there.
Yes. Thank you very much, Chris, for this very nice talk, which was very interesting, and all these final ideas for discussion. So if anyone has a question or just want to discuss, just raise your hand, or even you can unmute because we are not so many people. So yeah.
Hey, Sven.
Yes. Hi, Chris. Hi, everyone. Yeah, thank you very much for this great presentation, and also yeah, thanks a lot for this effort of like doing this conversion of databases and cell types and metadata. This is something, it's true, it's very important for the community, and I think it's going to be very useful to a lot of people. I was wondering, in your talk, when we do single cell analysis, there's always the question of when we do the differential analysis. Of course, there's some part of the biological reaction we are looking at that is due to changes in gene expression, and you've shown that on these the cell type, focusing on the differential analysis there, and also, of course, there's the difference in cell type proportions, relative proportions, and of course, sometimes the gene expression in one cell type is part of the response, and the difference in cell type proportions between both conditions is also a big part of the signal you want to capture. So I was wondering if you have intuitions or like general guidelines about what's the best to look at, if it's always easy to compare both, informations or sources. Yes, so.
Yeah, that's a great question, Sylvain. And nice to see you again. Yes, so I agree with that. And one of the problems in that area is if you cluster based on similarity of gene expression, do you mask the cell abundance differences? And I agree with that. One thing that we found is we've been using MILO-R, MILOR, and the advantage of MILO-R for predicting differential abundance of cell types. The advantage of MILO-R is it doesn't use the cluster information. So you may have clustered already, but MILO-R doesn't use that cluster information, and it uses a network approach, sorry, a neighborhood approach. So the neighborhoods are very small relative to clusters. So the neighborhoods are predicted based on similarity of expression, but the neighborhoods themselves are very small, and there's basically a cell that's the starting nucleus, and then it collects around that cell, and it iterates. And the advantage of that is that you then look for differential abundance within the neighborhood for your treatments. So say you have a specific neighborhood and there might be 200 cells in that neighborhood, and there may be a differential abundance between treatment, then you can do a calculation and show there's a differential abundance within that neighborhood. And then and that's independent of all other neighborhoods. So it separates the problem of trying to combine cell predicting how many cells you have from the gene expression changes. So I think that's an advantage. And then what you do, if you do want to try to understand what cell type is in that neighborhood, you can still say, all right, the cluster label can be merged back to that neighborhood. So neighborhood X, the cells in that neighborhood, of course, have a cluster identity and a annotation of that cluster. So you can still find that that neighborhood was a CD4 positive T cell, for example. So I think that I like conceptually that's good, and I'm the statisticians tell me that that's a robust statistical argument. So we're looking at proportions within neighborhoods, and then you can annotate those the cells in that neighborhood and so forth. So that's what we've been doing, and so we think MILO-R is a good way to separate those issues.
Right, thanks.
Yep.
Any more questions?
I do have a question, actually. So I work on cattle, and my specific topic is cattle genetics, but I'm kind of going towards selective breeding. And I was just wondering, since you showed us your single cell approach on the PRRSV resistant and susceptible pigs, how do you think this knowledge can be applied to selective breeding?
Yes, that's a very good question. I think that there's a couple of possibilities. You can take the human cell atlas approach, and so one thing that they've been doing is trying to use molecular phenotypes as alternative intermediate phenotypes as predictors of animal phenotypes. And so they're using EQTL analysis for that. And there's a consortium that is trying to take the next step and to look at single cell EQTLs. So the idea would be, well, there's a single cell EQTL gen. So that's the consortium. And Monique van der Wijk gave a presentation last week in the last field day, and that's recorded. So if you want to go on the website and get a 30-minute description of the single cell EQTL work that's going on. So that would be one idea. And I didn't put it in this presentation, but I've put in presentations in the past. The ENCODE project and several other projects have shown that the SNPs associated with human traits are often context specific. So pancreatic cancer associated genes are SNPs are found near genes that are expressed in the pancreas. So, the idea is that we can try to identify the most important SNPs by looking at the biological context. And they can show even more so that's true of the cell types within a tissue. So if you can find gene expression that's controlled by a SNP in a particularly important cell type, that may be more pertinent information for filtering SNPs and predicting those SNPs are more important than other SNPs that might be in LD with each other. So that would be, a SNP filtering approach. That's one. And then, as I mentioned, the other is because we may need to look at the single cell level, if we can look at gene expression itself as the phenotype. That's what we're hoping to do with one project where if you're familiar with the disease resilience project that Jack Deckers is running with Graham Plastow, who I think is on the call, yes. So Graham can tell me what details I'm getting wrong here. But essentially the idea is breeders can't directly measure disease resistance in their nucleus herds because, of course, we don't want to bring disease into the nucleus farm. So how do we come up with predictors in healthy animals that would predict phenotype in diseased, normal, common, economically important environments? And so the idea is, can we look at blood phenotypes? And so we're looking at blood gene expression. We're looking at other cell types. Other phenotypes, protein phenotypes in blood and so forth. And so again, connecting those two ideas, we may need to look at gene expression within specific cells in the blood to really find the most important SNPs. So that's the current approach that we're taking. Basically, can we find new associations? And can we use RNA markers potentially even at the cell type level as intermediate phenotypes to predict?
Thank you. Thank you so much. It was super interesting. And from this, I do have another question, which is, how can we improve bioinformatics tools to interpret these data and also kind of implement everything together to develop maybe some selection strategies, if you have any idea?
Yeah. Well, that's a very large question, and I can't answer that directly. There is a enormous field literally there's a new method published every day in this field. It's literally every day. And for example, if you look at the most recent papers, there was a big raft of papers that came out in the Human Cell Atlas community field just at the end of last year and even into this year. One I gave a reference to. There are lots of people working on that. I think where they're not working, where maybe we want to focus, and I would say, in addition to all the things that we have to do in terms of infrastructure and interacting with each other, I think, and building the databases, we're going to have to do that ourselves, because obviously the databases are not necessarily going to be the same structure on the human side. So we're going to have to do a lot of that ourselves. But where they're not really working at all is in the area of trying to use the types of data sets that we have. So, they don't have data sets on highly related, environmentally similar, but genetically diverse but related individuals. So they'll have completely unrelated individuals in very different environments, and that's the data types that they're using. What we would have, right, is in cattle, for example, there's an architecture, genetic architecture to your population, which can we use that? Could that be part of the analysis where can you use genetic relationships to help us integrate the data more easily, for example? Would we expect cells and the transcriptome of single cells to be more similar within cohort or family relationships? And would that help us with this problem of avoiding trying to have batch effects, but avoiding the loss of important differences that we want to follow? So that could be one thing that we work on. And this area of sampling across environments that in the human community they really don't do, where we might be able to look at, on the biological side, I'm really a biologist by training. I would be interested in, can we take time points and develop better tools in terms of repeated sampling, which is very poorly addressed in bioinformatics and statistical genomics in the single cell field, because they just see that kind of sampling in humans. They do in mice, but it took us a long time to really identify pipelines and packages that can handle things like repeated sampling and repeated measures as a random effect. And we're currently using a software program called MAST, M-A-S-T, which appears to be pretty appropriate for that kind of analysis. But those would be the first things that come to mind. It's a huge field, and I'd encourage you, if you're really interested, try to get to Orlando, and we will try to have this discussion there. It will involve also plant groups, but I think that's one of the things that we want to consider, is how can we work as an agricultural community to at least the front end. Can we help each other collect data? How is the data? Describe the data, and come up with tools to share the data, that it doesn't matter what species it is. That would be the other, getting involved on the front end, so that the data is available for us to use.
Thank you.
Sure. Other questions?
Yes, well, I have one, Christopher. First of all, congratulations, fantastic work and very nice presentation.
Thanks.
My name is Inaya. I'm actually not exactly from this field. I'm a geneticist. I work with genomics and animal breeding here at Purdue University. And well, I'd say that my work is mainly focused on bioinformatics and statistics. So I have been, taking some challenges nowadays, and I have been working in the convolution analysis. That's why I started to work with single cell as well. And one thing that, things are working okay, but like I said, it's everything completely new for me. So I truly appreciate your talk today. I learned a lot. And one question that I have for you, because this is quite related to things that we are seeing here, is what is the impact from different breeds that you expect in the analysis, right? Because we are seeing some difference depending on the reference data that from the single cell data that we are using as reference in the convolution analysis, we get slightly different outputs, right? So it seems that this relationship between the breeds is important. So I assume that for us working with livestock, different species will likely have an even higher impact, which is we don't deal with that in humans, right? Do you have any thoughts on that?
I have some thoughts. I don't know how well they're grounded in experience, because we're just starting to try to think about that. But there's clearly evidence that genetics is controlling gene expression even at the level of individual cells within the same individual. There's some differences due to allele bias within a single cell, and you can see that. So I definitely would expect breeds to have influence on the gene expression pattern. I think that the way that the human community is starting to think about that, for example, in different human ethnic groups, they're starting to deal with that as an additional factor. And the question will be, are there going to be different reference maps for specific ethnic groups, or will those be interpreted as variation within a cell type? It's unclear. They're not really any further along than starting to develop these databases, just collecting all the data. And I understand your questions. I think that there's going to be that we will also have to deal with. The issue is, and it's becoming a more philosophical question on the human field, is what is a cell? I mean, how do you define a cell type? What are the factors that might change a cell from a particular cell type to another cell type? And genetics would be one. What we're doing right now is trying to define cell type at a certain level, and then argue that within a cell type, there'll be subtypes that you can identify by transcriptomic pattern, and we usually call those cell states. So we have cell states within cell type, and the question is are those cell states specifically controlled by some other factor, and is that other factor the underlying genetic variance? I think the problem is you won't be able to answer that question until you have hundreds or thousands of individuals. And that's one thing that the single cell EQTL gen consortium is set up to do, because they are collecting whole blood, making PBMCs, and predicting gene expression and cell composition of thousands of blood samples from different ethnic groups. So I think they're just started. They only have a few initial papers out on how they're going to do things. But I think that would be something to really watch for, to see what the level of difference cells are from different ethnic groups. And maybe we'd be able to apply those same ideas across breeds.
Perfect. Thank you.
I have a question. It's related to your previous comment about joining the animal and the plant community under this umbrella of the agricultural community. And this is actually something that we were discussing when we started the group. We were not sure whether we should name it animal genomics or something that actually was both for animal and plant genomics. But we decided at the end that as these communities are, yes, they are quite established, and the genomes are different, so plants can have polyploidies and more repetitive reads and stuff like this. So at the end, we decided to call it animal genomics, but maybe we can rethink about it. So how do you think that these two communities can actually be reconciliated and actually join efforts for this kind of creating these databases, or at least to have this kind of methods that both communities can use?
Yeah, I mean, that's something that we've been thinking about as part of this AG2PI consortium. I think that there are two areas that what I would call is low-hanging fruit. One, areas that I think there's not really any biological or discipline-specific roadblock to working together. And one is the front-end data analysis. So analyzing or collecting the data, is going to be discipline specific or species specific, often because of the differences between the organisms. But once you have the data and housing it, sharing it, and initial analysis of it, I think those are places where we can work with the plant community very easily. There will be certain descriptors that are going to be different. They have a plant ontology. We have an animal ontology that are different for cells, are different for describing the samples. But that's really something that each community can work on. And then depending on what type of sample it is, you just crank through that pipeline. So I don't see that as an issue, although it's not something that we work on together. But building the warehouses, building the tools to access the data, and maybe initial analysis, maybe even to the point of creating reference databases. I think those things are things we could work on with them easily. The other major area is training of people to use these tools and to analyze the results in at least an initial way before you get to field expertise. I think training people to use these tools that I just described is definitely something that we could develop much better. There's interest in that in ag biodata, the consortium, that as well as in the plant cell atlas community. And there is a plant cell atlas community. They have National Science Foundation money to meet. And they have a big white paper, an excellent white paper on how single cells can be used in plant biology. And I'd recommend you look up Plant Cell Atlas if you're interested in that. But I would say at least those two areas. Any kind of data processing and analysis that doesn't involve field-specific information and training of the people that we need to move forward with this.
Okay, thanks. Is there any other question? Okay, if there is no more questions, thank you everyone for attending the seminar. Of course, also thank you, Chris, for the nice talk. And I would like to announce that for April sessions of the group, we will have as a speaker Damir Barnasik from the Imperial College of London, who will discuss about the NeoCode project, which is an international collaborative effort that aims to annotate the functional elements of zebrafish genome. And yes, just to mention also that next week is the NFCore Hackathon. It's an online event, also with local sites, but the registration for the local sites are already closed. But if any of you is interested in joining the online hackathon, you are more than welcome. So thank you, everyone, and bye. See you in another session.
Thank you.
Thank you. Bye.
Bye-bye.